So just a few slides to introduce what we believe are the relevant changes or clarifications in the overarching guideline. We have tried a definition of biosimilars, and I can tell you this is not an easy task to do, because although you think it's easy, there is always there is hardly any definition that makes everybody happy. But now we believe we have found one, and we can of course discuss this. And the definition is as follows: a biosimilar is a biological medicinal product containing a version of the active substance of an already authorized original biological medicinal product, the reference product. And then we go on to say that how a biosimilar demonstrates similarity to the reference medicinal product. I think a really large step forward is the intent to facilitate global development of biosimilars. Now that it's possible from a legal aspect, we of course have to define the science and to agree on the science, how we could really facilitate that global development. In the overarching guideline, we give some guidance to follow on how this could be done. So we say that a non-EEA or EU authorized version of the reference product may be used for certain clinical and in vivo non-clinical studies where they are needed, if this reference product is authorized based on similar scientific and regulatory standards as in the EU. So we are thinking about the ICH regions here clearly, and if that material is representative of the reference product in the European Union. And for that, actually, applicants or sponsors will need to provide some bridging data, some solid bridging data. And these will typically include a three-way analytical comparison with regard to structural and functional characteristics, and may also include a three-way PK and or PD comparison. We also try to really tailor the scientific program, the development program for each biological, because we know that they are not all the same, and there are rather simple, maybe polypeptides, and they are very complicated biologicals. And what is new in the guideline is that we say in specific circumstances, a comparative clinical efficacy study may not be necessary. And this is true when similarity of the physicochemical characteristics, biological activity of the biosimilar and the reference product can already be convincingly shown, and similar efficacy and safety can clearly be deduced from these data and comparative PK data. So that would be more the generic approach, although of course with generics you know that we don't have a comparative data set of the quality characteristics. Such an approach, however, may have to be supported by additional data. So I think that's something new in the guideline. Some clarifications are given that we believe were necessary. We believe, or we think that the scientific principles of a biosimilar comparability exercise actually are based on the same principles for evaluation of the impact of changes in the manufacturing process of a given biological. While we could discuss, of course, that the extent of whether, while it is probably clear that the extent of data might differ, we still believe that the scientific principles are the same. We also highlight in the guideline that intended changes to improve efficacy are not compatible with the biosimilarity approach, because they might, for example, you change the posology. You might need different doses than for the reference product. However, if you have safety advantages such as lower levels of impurities or reduced immunogenicity, then such differences should be explained, but they may not preclude biosimilarity. We have put that in the guideline because obviously it was not self-evident, and we have been asked these questions over and over. We also try to clarify that EMA evaluates biosimilar medicines for authorization purposes only, and that it's not within the remit of the EMA to evaluate interchangeability or even automatic substitution or to give recommendations on these issues. This is clearly within the remit of the EU member states.